Hi all! Welcome back to Hology. I am Nucleic. Today we are going to discuss about cholera toxin and its mode of action. Let's dive in. Epidemic severe watery diarrhea which might lead to death if untreated. The causative agent Vibrio cholerae secretes a potent enterotoxin called cholerae.CT, which is largely responsible for the disease. Robert Koch, a German physician and microbiologist, was the first person to postulate the existence of cholera toxin. In 1886, Koch proposed that Vibrio cholerae secreted a substance which caused the symptoms of cholera. Koch's postulation was proven correct by Indian microbiologist subunit.The. concluded from this experiment that an endotoxin liberated upon disintegration of the bacteria was the cause of the symptoms of cholera. The organism colonizes the small bowel and secretes a protease which activates cholera toxin or CT. CT consists of one enzymatically active A subunit and 5 immunogenic B subunits. The B subunits form a symmetrical pentamer into which a chain sits on. The A subunit of CT is non-covalently associated with the B subunit pentamer via the disulfide-linked C terminal at 2 fragment. When cholera toxin is released from the bacteria in the infected intestine, it binds to the intestinal cells known as enterocytes through the interaction of the pentameric B subunit of the toxin with the GM1 ganglioside receptor on the intestinal cell, triggering endocytosis of the toxin. The endosome is moved to the Golgi apparatus and to endoplasmic reticulum where the A1 protein is recognized by the ER chaperone, protein disulfide isomerase. As the A1 protein moves from the ER into the cytoplasm, it becomes an active enzyme. The subunit of CT modifies a membrane-associated protein called a G protein, which binds to the enzyme adenylate cyclase and controls its activity. So first we will take a look at the action of GPCR. G-protein-coupled receptors are cell surface receptors that pass on the signals that they receive with the help of heterodimeric guanine nucleotide. Binding proteins, also known as G-proteins, alpha, beta, and gamma subunit. In an inactive state, alpha subunit is linked to GDP. When a signal molecule binds the G-protein coupled receptor GDP in alpha subunit is replaced by the GTP and it's separate from other subunit. The activated G-protein bind and activate adenylate cyclase, which converts small amount of ATP to cyclic AMP. Cyclic AMP activates protein kinase A, which induces numerous cellular responses, including the opening of ion channels include CFTR, cystic fibrosis transmembrane conductance, regulator, pass the chloride ions out of the cell. G-alpha subunit has GT-PACE activity. Under normal circumstance after a while, GTP is hydrolase to GDP. G-alpha recovers with beta and gamma subunit and adenylate cyclase is deactivated. The cyclic AMP production is also stopped. So the signal is turned off, and the cell returns to its equilibrium state. So what does this do with the cholera? The activated a chain of cholera toxin covalently modifies the G protein and traps in its activate state. This leads to constant activation of the pathway. The production of excess cyclic AMP and constant activation of pKa leads to loss of chloride and sodium ions from the cell. To maintain osmotic pressure, a large amount of water is lost as well. This large influx of ions in water and blood causes diarrhea. This helps the bacteria to disseminate in the environment. That's all, my friends. I hope this video is helpful. Don't forget to like, share, and subscribe. Bye-bye.